Hi everyone, I'm Misha and I'm a member of 2025 iGEM team from Maastricht University, Corspin. Within our project, we're introducing the iGEM interview series where we want to bring the state-of-the-art synthetic biology closer to you. In each episode, we sit down with leading scientists, engineers, entrepreneurs to explore their groundbreaking research, novel applications, and the thoughts that are shaping the synthetic biology of tomorrow. Please, let's dive in. Hello, George. It's truly an honor to have you as our guest today for our iGEM interview series. I'm Misha, MSP student focusing on synthetic biology. And thank you very much for joining us.
My pleasure.
So first, let's go back to the very beginning of your scientific career. You entered your bachelor's at Duke University and completed a double major in zoology and chemistry in just 2 years. That's unusually rapid start. So why and how did that happen, actually?
Yeah, so I had some advanced placement from high school, and I didn't want to spend a lot of money getting a undergraduate education. I wanted to get into graduate school where they would pay me to do research, and I basically wanted to do research.
Mhm.
I didn't, I figured I would learn as I went along. So it
Yeah.
didn't feel like rushing. It just felt like getting to the next stage.
Oh, okay. Okay. All right. All right. So and basically these in these 2 years. Um, did it yeah, like I would imagine for us as an undergraduates, you get your the very beginning, the very first touch on your on your scientific career at that time. So did it shape your outlook in science in any way?
I had done research in high school, independent
Mhm.
research, where I had keys to the chemistry lab, but uh, I did. I really wanted to get into. I also did research as an undergraduate in and
Mhm.
around the courses. Um, I took the lab. Some courses had lab versus course option, and I would always take the optional lab course, even though it didn't affect the grade.
Mhm.
And also would take graduate courses, and I worked in a lab for a salary as well.
Mhm.
I was getting research from a lot of different angles during those years, but it transformed another notch when I
Mhm.
finally got to do full-time research in graduate school.
Yeah. So now talking about the graduate school, at Duke, as I read, you dedicate a lot of time, like even you set up to 100 hours to research, so that you didn't really care, didn't really focus on the coursework, and you were withdrawn from the program. So, but you completed your PhD at Harvard with the Nobel laureate Wally Gilbert. So, um, what did you actually learn from this setback in Duke?
Well, I never really felt it was a setback. I just
Mhm.
kind of just kept doing stuff. The reason I had gone to graduate school is so I could be a full-time researcher.
Yeah.
And then they told me I had to take the courses that I had already taken, or equivalent courses, and I just said, no, I already took those as an undergraduate, same school,
Hmm.
same professors. And so I said, I want to do full-time research, and I did it. And then when they dropped me out of the program, then I just did full-time research as a technician. And
Yeah.
then my advisor, wonderful advisor, Sung Ho Kim, said, you really should try PhD again. And so then I fortunately got into Harvard. It was the only place I applied. And, but anyway, um,
Mhm.
then I had to take courses again for the third time. But this time I said, okay, I will spend some time doing the courses.
Yeah, that's fair. And also, also talking a bit more about the Harvard time. Like Gilbert's lab is just an amazing place, I would imagine, right? With co-invention of the first DNA sequencing with Zanger and everything the novel techniques. So, what was it like to be a young researcher with in this era of the breakthrough technologies when we got the first sequencing methods and basically the modern biochemistry and the molecular biology was developing.
Yeah, well, molecular biology was being turned into a technology. It was
Yeah.
not just reading DNA, but it was also writing it. So it was the whole recombinant DNA was happening all around me. Uh, we, it really laid the groundwork for next-gen sequencing and synthetic biology, two things I was going to um, really focus on in the future.
Mhm.
I mean, it wasn't obvious, but it was. But looking backwards, you can see how it could have helped think about it.
Mhm.
It was just, and it wasn't just Wally. I mean, we were surrounded by very talented um, students and professors. You know,
Mhm.
Tom Iotis and Mark Potashny and Nancy Kleckner, um, Matt Esselson. These were some of the ones labs that I rotated in.
And if you had to say the most important thing you learned from all of these very, very good researchers at that time, what would it be? One thing.
Well, from Wally, you know, I got reinforcement of two of my natural tendencies, which is towards technology development and um, being able to work in multiple fields at once, sort of the sort of uber multidisciplinarity. He
Mhm.
showed that that could be made to work. Um, I'm not sure that he or I did it completely consciously, but it was very reinforcing. Um,
Mhm.
Boldness, you know, don't do something small. You know, it's going to take almost
Mhm.
exactly the same amount of time to do something big. So, yeah, those are some of the lessons. Yeah.
Nice, nice, nice. And jumping forward to 1984, at a meeting with Department of Energy, you and also some scientists proposed sequencing an entire genome. Like basically, the idea of Human Genome Project was was first established. So, how did you and your colleagues manage to launch the Human Genome Project?
Well, it kind of fell in our lap, but in a certain sense, it was kind of an accident. The meeting was supposed to be on estimating mutation rates in populations, and we felt we couldn't do that
Mm,
without at least without a reference genome to say how they were different. And
mm,
so then we had all kind of been independently thinking a little bit on the edges of that, but we kind of all put it together and said, well, we
mm,
could do a genome, and then we described how we would do it, and then that went back to Department of Energy headquarters, and they
mm.
thought it was great. And you know, there are only about 10 scientists in the room, and you know, a couple of months later, they were writing checks. To get the Genome Project going. So that was 1985-ish, and it really was going quite well under the Department of Energy with you know technology development,
Mm.
you know, and you know computer robots, all kinds of things. And then NIH got in the game because they didn't want DOE to be responsible for healthcare and or responsible for the greatest breakthrough in healthcare.
Mhm.
And then I think it went downhill from there for a while, and that it
Yeah.
just innovation was downplayed and production was up. And then it wasn't really until 2004 that victory was declared before it was actually done, and that allowed us to go get back to doing technology development. And almost overnight, as soon as they raised the
Mhm.
allowed the door to open. We got to next gen sequencing, which changed it from a $3 billion project to a $1,000 per now
Yeah.
$100 project.
I just wanted to ask you about this actually. So, um, how did that happen actually? It just if we think about it, this is just insane, right? So something first it cost like $3 billion, as you mentioned, and then it goes down to a couple of $100. Yeah, like
And the
it's
quality went up too. So it
yeah.
wasn't just bringing down the price. It went from 3 billion base pairs, which
Mm,
isn't even a practical genome in a
mm,
clinical sense. You need to have both your parents' contributions, which is 3 billion
mm,
each, is 6 billion. So it should have been twice as big, and it's actually harder to separate two genomes that are that closely related and
mm.
tell which one came from the mother and father. All that's important. So it's a very low quality genome in every way, and it had lots of gaps in it. Anyway, now we can make nearly perfect separation of maternal and paternal chromosomes.
And looking ahead a little bit, how would you see maybe these technologies integrating in the routine healthcare? For example, we now take blood tests or all kinds of other tests, but would maybe doctors in the future be also looking at your genome when yeah, when you just go for a checkup?
Well, certainly in the future, and to some extent already in the present,
Mm,
a large fraction of cancers should and are there. You do genomes both preventatively and also of the cancer itself if you've already got cancer.
and also, as a next step in the Human Genome Project, in 2005, you launched the Personal Genome Project to sequence the genomes of different people and make the data actually publicly available. And at that time, and I feel like we still are worried a bit about genetic privacy. So, what actually motivated you to push for open access personal genetics?
Well, it wasn't that I wanted everybody in the world to forget about privacy. I think it was more a matter of if we were going to be sharing the data, which
Hmm.
everybody wanted to do, we should properly consent people. We should allow them to look at their own data, which was not possible before my project. We should
Yeah.
allow them to share their own data if they want to. Um, but most importantly, we should communicate to them not just the possibility, but the likelihood of it escaping. While most consenting at that time was said, we can't tell you what we found, uh,
Mm.
and we will keep it secret from everybody else. When in fact, what happened is because of hacking and you know lost disk drives and whatnot, it could be that lots of people know your data and you don't. So it was really my reaction to human subjects consenting. Now, I think in terms of privacy, I think now we can make, with things like homomorphic encryption queries, we can ask questions about genomes without decrypting them. So if they're
Mhm.
encrypted, you can make it so that even that nobody can read it. They can ask, a very limited set of questions about it.
Yeah. That's a fairly good point. Yeah, a lot of yeah, but still, it's yeah. I guess it's not it's a problem of communication of these technologies a little bit because yeah, because I feel like yeah, when I prepared for the interview, I asked my parents like. Yeah, like what do you think about the Personal Genome Project? Would you like to potentially maybe share your information with yeah, like with the scientists? And yeah, and they were also kind of skeptical. So do you think it's how can we properly no, how can we better communicate this to a wider public so they're kind of more prone
Well,
to
again, the objective was not to get 8 billion people to share their data.
yeah,
It
yeah.
was to test new technologies, including
Mhm.
multi-omics technologies, on a cohort that knew what they were getting into and could prove
Yeah.
that they did by getting 100% on an exam that showed that they knew what they're getting into. It's going to be a tiny subset of society, but it could be, as far as I can tell, there's a diverse, there are people from almost every ancestry that are willing to do this.
Yeah.
You don't need everybody. But furthermore, the sharing question is not whether you're going to share it with scientists, it's whether you're going to share it with hospital staff.
Yeah, with the hospital.
Okay.
Yeah.
Scientists, for the most part, couldn't care less who your name is, but how hospital staff, if
Yeah.
if a celebrity comes in, they're gonna poke around and find out what's going on, which they shouldn't be, but they do.
Yeah. Yeah.
So I think that people do share all kinds of medical data, not just
Yeah.
sequences. And the
Yeah.
medical data is actually, in a way, more personal, more
Mhm.
private, than your genome, because your
Yeah.
genome might say that you're at risk for Alzheimer's. Basically,
Hmm.
everybody is if they live long enough. Uh, but your medical records will say, oh, you have STDs and you have,
Hmm.
early onset dementia and
Hmm.
et cetera. So anyway, I think it's a personal decision when we do need to communicate
Yeah.
um, better. It's interesting that people aren't getting their own. It's not a matter of sharing the science. They don't even want to share it with themselves. They don't want to get their own genome sequenced. Uh,
Hmm.
when it could save trillions of dollars, it could save, um, personal, family anxiety
Hmm.
and you know, s- if you have someone with a very severe genetic disease, then somebody in the family often has to quit their job. Sometimes
Hmm.
both of them, both parents. Anyway, uh, there's going to be a lot of education in the future, and there are many technologies that take a while for people to understand what they're good for. For example, computers. When I first
Hmm.
asked, when I was young, I thought computers were great, and I asked people if they wanted them, and they said no way. And now you can't get it loose from them, so, so it's
Yeah.
just a matter of seeing the benefits and you know,
And just yeah, and just evaluating for yourself what's good and what's not, and maybe for the bigger good.
yeah.
Yeah, yeah, and also talking more about the yeah about more applications of DNA. You've also used the DNA for data storage, and famously you encoded your full entire book, Regenesis, with more than 300 pages with images into synthetic DNA. And it's just insane. Just a drop of this DNA liquid, as I read, it contains 70 billion copies of the text. So, what actually inspired you to do this experiment, and how did you actually do it?
Yeah, so you know, I had been working on reading and writing DNA basically since I was
Yeah,
a graduate student that we talked about earlier, and it just
yeah.
got exponentially better and better every year. And then one year I realized it was good enough that we could store a book in it. And and I wrote a review for uh, an article that I wrote it in DNA, and the editors let it go through. So I had the software already done just for that
Mm,
stunt of reviewing an article on synthetic biology and DNA. And I so it was fairly easy. I mean, it was just we used two of the techniques from the lab. One was to write it using chip-based synthesis, which
mhm,
we had published in 2004, and then amplify it. So actually, one of the advantages you could make billions of copies almost for free for pennies, um,
mhm,
and then uh, and then to read it with fluorescent next-gen sequencing, which was one of the two main methods that I had helped develop. The other one being nanopore, which
mhm,
wasn't quite ready at the time. This is around 2014. Now it is totally
mhm,
ready. Um, yeah, and then compare the input with the output and saw that they were basically the same. We had error checking capabilities, which made it so you really didn't have to have very accurate reading or writing because of the
mhm,
error correction.
mhm.
But
Okay.
it was on the order of 99+% for both reading and writing. Raw data, that's just for one read, and then the redundancy went up, that got
Mhm.
exponentially more accurate.
That's insane. Insane. Yeah, and do you think for the real world use, yeah, like we talked about this, right? This all happened within the university, within the lab, but do you think that this kind of DNA storage can become practical for real world use?
So outside of academia,
Mhm.
it's mostly outside of academia, after that paper, and where there's an international consortium that's trying to do this. I think the
Mhm.
first applications are not going to be what the top priority of that consortium, which was essentially writing digital data into DNA. I think it's going to be writing biological data into DNA because it's a easier route. It's a much cheaper route. You don't have to go through. You don't have to go through the interface between biology, between digital and biological. And if so, for example, we and others have demonstrated you can record all kinds of developmental lineage data and
Mhm.
physiological data into DNA and RNA and,
Mhm.
and even proteins, and then you can read it out in a way that, that where you go to a particular part of the body and you know that that data was from that body type and you can body part, and you can get a time record and so forth. So I think that's a that's the lowest hanging fruit. It's not that low hanging, but it is lower hanging, I think, than trying to compete with disk drives. I think for a
Mhm.
while, so you can get potentially exabytes of data in a living system,
Wow.
while writing exabytes with the digital to biological route that we described in my first paper is still out of reach.
Mhm.
But doing it entirely biologically, you can record that level, and then you can do selective retrieval and sequencing.
Yeah, and also over past few decades, you've been working on an astonishing range of biomedical advances, everything from the Human Genome Project that we talked about to CRISPR gene editing and also personal genomics, right? The age reversal and the de-extinction of animals. And my question is, how do you decide? So this is the next biggest thing that I want to work on, especially when when these areas might not have immediate payoffs. Like,
Yeah, well, it's been my experience that things tend to pay off faster than you expect rather than slower in this particular
mhm,
set of fields. These are exponential fields. And so people look at the short term and it seems very slow, but just even a moderate term, the exponential is surprising. And that's computing and AI and reading and writing DNA, and then cellular, anything that comes from reading and writing DNA, all is gone exponential by
mhm,
10 million fold or more. So that's so the way you pick then is you can either go forward where you say, what combination of new methods can be put into an even newer method. So like we just went through taking
mhm,
reading and writing DNA and integrating them, or
yeah,
you can take cell biology and barcoding, the multiplexing that made both reading and writing go exponential. You
mhm,
can multiplex, um, you know, of viral delivery, vectors development. You can multiplex cells, where you can do millions or billions or even in some case trillions of assays all in one tube.
yeah,
And then you just you just report the winners. You can't necessarily sequence trillions in every case, but you can pick the winners and sequence those. So, so there's so you don't need to see the immediate payoffs,
mhm,
although I think in general we have. Some things were faster than others. So, next-gen sequencing was picked up much faster than, say, fluorescent next-gen sequencing was much faster
mhm,
to pick up than nanopore. Uh, CRISPR was uh, much faster than de-extinction.
mhm,
Both required editing. We would kind of collect ideas for why you would need multiplexing. It was kind of multiplex was driving this,
mhm,
both the radical reduction in cost and improvement of quality, and we'd say, well, we need multiplex editing for recoding genomes, for humanizing, for the extinction of alleles and so forth, and then we would just go through and go and try to make them happen.
So do you plan on revisiting maybe the like the original genome project, and you're still continuing, right?
Oh yeah.
So
So I mean, we read, like with most of these exponential technologies, as soon as it becomes available, you can redo everything that's been done in a short period of time inexpensively. So for example, in 2009, we published a paper where we had the Genome Project was just finished in 2004. 2009, we did 5 genomes, diploid genomes, so they were twice as big, so really 10 times as much data with higher quality. And then today, we are aiming to do, all 8 billion, or at least the subset of the 8 billion that want to have better healthcare. And you can see it happening in places like UAE and the United Kingdom and Iceland. UAE is getting close to getting its whole population sequenced.
wow.
And United Kingdom is close to having the same number of people, but it's a smaller fraction of their total population, on the order of a couple of million people. But it's going to be billions if, assuming people see the health.
The practical use, basically.
Yeah, health uses and financial benefits. Yeah.
And now going to another area of your research, CRISPR-Cas9 systems. So and CRISPR is definitely a revolutionary technology, but the technical complications currently, like off-target effects, are still being mitigated, right? And the delivery methods of the CRISPR therapies are still haven't been worked out fully yet. And do you actually think that we are close to solving these issues now with the new technologies that we have, and with with basically all of the stuff that we're doing now with AI, maybe?
Yeah, so we were working on other editing methods before CRISPR, and we're working on other ones afterwards as well. I think that we were the first one to raise the issue of off-target, I think. But it hasn't been that big a problem, really. Just briefly, there was a off-target problem with C-base editors, but that was quickly solved by David Liu's group by adapting the A-base editors to Cs. But the vanilla original CRISPR, in 2013 that we published, pretty much had such low, if you choose your sequences carefully, could have very low off targets. But I think multiplexing is hard. That's doing lots of edits. I think the record now for CAR T multiplexing is on the order of 6 edits at once that came from our group at Kihan. But if you really want to do higher levels, then germline is a good way to go, and we do that in pigs, where we can edit up to 69 edits and probably more in the near future. And you can make them almost perfect because you're doing clonal, so you have one cell that makes the embryo, and so that means that you can make that cell essentially perfect, so that you can sequence the whole genome and make sure that what you have is correct, you haven't missed anything you wanted, and you didn't get anything you didn't want. So I think that's, and that kind of, even though it's can't do germline in humans yet, you can definitely do germline in pigs, and then that makes its way into humans.
Yeah, that's true. That's true. And also, sticking to this topic, you have you have introduced, as you said, 69 or more gene edits in pig cells. And the goal was to inactivate if I understand correctly, right? The goal was to inactivate the retroviruses in the pig genome for basically making the organs from pig transplantable to humans. So specifically on that project for organ transplantation, how do you think, how close are we now to normalizing? Yeah, because I've heard that there was a story in China in 2024, the first liver from the pig that like with I believe with your technology was transplanted into human. How do you think? How are we close to normalizing this transplanting gene edited pig organs into humans, like on a normal basis, basically in each hospital to end the organ shortage?
Right. Well, it's more than just the organ shortage. We can make organs that are better
Yeah.
than organs in principle. Things that have better immune profiles that are not rejected um, the way human to human organs are occasionally rejected. We can make them so that they're pathogen resistant. In fact, pig organs are intrinsically resistant to all human-specific pathogens, things like, the syphilis bacterium and HIV virus and so forth. Um, but anyway, the way that it gets, we do both have
Mhm.
better organs and solve the crisis of shortage. Uh,
Mhm.
it's I mean it's already happening. We've transplanted 2 kidneys now and both people are still setting records for
Wow.
longest survival. So one of them is now 7 months out and is pretty healthy. I mean, considering that they had, in addition to the kidney problem, they had other health problems because that's how they were prioritized to be
Mhm.
involved
In the study.
in the study. But as we get to healthier and healthier patients where their only problem is kidney patients, I'm sure it'll go better and better. But already these two are doing well. And kidney is probably the biggest unmet need right now worldwide, hundreds of thousands of people. Um,
Mhm.
so normalized, I mean, I think this, unlike getting your genome read, which seems to require some explanation as to why it's useful.
Yeah.
This is obvious to all the patients. Almost everybody on dialysis wants to get off dialysis.
Mhm.
They really. While I think, and I think the difference is one is 100%, guaranteed to be better. Dialysis is,
Mhm.
you have brain fog, you have this really inconvenient, like constantly going to the clinic, kind of limits you geographically. Um, while with genetics, it's like, well, there's a 97% chance your kids are going to be fine or your first child will be fine.
Yeah.
And so in one case, it's 100% chance you're going to have better outcome if you do the transplant. And in
Mhm.
other case, it's like maybe a 3% chance that you'll benefit from it. And I think that's what people can't wrap their head around. But from a public health standpoint, it should be obvious, and it will be.
Yeah, yeah, I definitely hope so as well. Yeah, it's insane how much we can achieve with yeah with the power with the powerful tools in our hands, you know.
Yeah.
Yeah. So you've also often discussed how gene drives could potentially spread genetic threats through wild populations. For example, yeah, making mosquitoes resistant to malaria, or yeah, or eradicating the invasive species in the regions of the world where they just are, yeah, are basically harming the native habitat. So this is actually a very powerful, I think, but also a bit controversial tool. So do you think we're close to see the gene drives actually released to the wild? Yeah, maybe for example with with a mosquito to wipe out the mosquito-borne diseases.
I, it's hard to say, because this is not a technical question. This is a
Yeah,
societal decision.
yeah,
Uh,
yeah.
I think that it might happen first with Lyme disease, and the solutions might be broader than gene drives. I mean, there are
Hmm.
ways that you can kind of, for let's say an island, you can completely saturate it with engineered mice,
Hmm.
not in a way that it spreads like a gene drive, but a way it spreads like regular genes.
Hmm.
Um, But anyway, one way or another, Lyme disease, malaria, one of them more of a rich person's disease, but I think they all could benefit, and this is something that's incredibly cost effective. It's just we need to do it cautiously. It can be tested. There's some large domed villages that have physical containment.
Mm-hmm.
Uh, we have also published papers on how you can do biological containment by having things like daisy chain gene drives. Uh,
Mm-hmm.
but yeah, I think it's worth considering. It's
Mm-hmm.
worth doing experiments cautiously. It could have an enormous impact, especially on impoverished regions where people just spend a huge fraction of their
Mm-hmm.
time, money, limited money, and health dealing with preventable diseases.
How do you think what should we consider actually before letting these, like, I'm not only talking about mosquitoes but also just genetically engineered animals. What do we have to also consider before letting them go to the wild?
Well, I think that most of the complaints about genetically engineered organisms, GMOs, have had to do with food,
Mm-hmm.
because the benefits were not evident, and so even though the downsides were also not evident, there was no reason to take any chances at all. There was just
Yeah.
no motivation, right? And so and even for the farmers, it was a small financial motivation. It was so that was with food. But with malaria, you're talking about 600,000, maybe a million lives per year that are affected, and that's 600,000 deaths and millions affected, and the same thing for Lyme disease and a few others. So I think that's the difference, and I've noticed a great deal more uh, enthusiasm
Mm-hmm.
or lack of antagonism uh, worldwide for things where it's very clear the health issues or the environmental issues. If you're talking
Mm-hmm.
about climate change,
Yeah.
genetically engineered organisms might help with climate
Mm-hmm.
change. There's a great deal of open-mindedness um, that rather relative to food, and also the food thing was not just very low, very low levels of obvious benefit. It was that there was this monopoly, Monsanto, that nobody liked, that they didn't do their homework well, and so forth, didn't listen. And now it's not just an American monopoly. It's actually a lot of contributions from Europe and other countries. For example, one of the inventors was from Europe.
Mm-hmm.
So I think that shared sentiment and the much more obvious enemy is going to change the conversation. But it's just a matter of you know
Public perception.
public and time. Time passes, and the technology becomes more and more evidently safe on the whatever applications are done. Like insulin is the oldest one. It's been produced in bacteria, which is clearly not natural, but nobody's going to take that bacterial produced insulin away from almost everybody in the world that has
Mm-hmm.
diabetes, that has access to that insulin now.
Yeah, definitely. Yeah, synthetic insulin production and maybe like yeah, maybe devices that help you produce the insulin by, yeah, like in your own body, like regenerative medicine solutions. I think it's the future. Yeah. And also uh, and also beyond the medical part and de-extinction and all the other things, you also applied synthetic biology to environmental and energy challenges. So you co-founded companies like LS9 and Joule Unlimited to engineer microbes for biofuel production and basically green chemistry. So it is definitely clear that biotechnology is a major driver towards a more sustainable future. But in what ways do you think biotech can have the greatest impact on yeah, on climate change and all of the pressing issues that are associated with it.
Well, some of it is, of course, speculative because we don't. If we had a solution to climate change, we would be deploying it faster than we are now.
Yeah.
And that applies to every climate change. I mean, most of the climate change solutions are just aimed at reducing human waste a little bit,
Mhm.
and that's probably not going to do it because we're way beyond the point where we thought we should stop, and so we have to do reversal, and that's a very different from just tightening our belts and not using our SUVs.
Mhm.
So so one speculation is that we can work on the Arctic, which has the best carbon sequestration in the world. It has 500 meters of topsoil rather than 1 meter in most of the
Mhm.
forests. And so we want to preserve in a frozen state all that carbon, letting go the carbon dioxide and methane, and encourage the better fixation that occurs in the more fertile and grasslands of the past. So that's one speculative thing. Another thing is it looks like there are mechanisms for fixing carbon, some of them not even photosynthesis, but they're all biochemical, for fixing carbon that could allow us to shrink our farms by a factor of 100, and
Mhm.
maybe even put them in people's kitchens. So that would eliminate the supply chain issues, and all you need is an abundant source of energy, which we're starting to get in the form of, you know, uh, um, you know, fusion reactors are not that far away,
Mhm.
windmills, and geothermal. So anyway,
Yeah.
that's a whole another revolution right there. So those would be the two big ones. One is carbon sequestration,
Mhm.
mostly in the Arctic, because it's hard to do it on farms or in the desert or in the ocean. But I think the Arctic is a great place, 19 million square
Mhm.
kilometers. And then the second one is reducing the farm so we can return the farmland to more natural and scenic
Mhm.
uses.
Yeah, yeah, yeah. Oh, that's a very good point also about the farms that you brought up also, like that we can actually put the farm potentially in the future somewhere, like in our kitchen, something like this. So are you talking about bioreactors, like growing the
Bioreactors with a diversity, a high programmability for
Mhm.
taste and textures and things like that, just in time manufacturing. And if you have to lock down, if there's some pandemic that's even worse than the last one, uh,
Mhm.
it's much easier to lock down if you don't have to constantly go out and get groceries. Yeah.
Yeah, yeah, you have a constant supply of food chain in your just standing near in your kitchen. Yeah, yeah, that would be actually amazing. So and also now going to your uh, yeah to your career as a professor. So a lot of people that are working in your lab mentioned that you have zero organization, yet they are shining with excitement, and they say that this is a place where you can either sink or swim, like a lot of freedom and a lot of exciting things you can come up with this like in this environment. So how do you actually create and manage this environment of so-called controlled chaos?
Yeah, obviously our goal is not to have anybody sink. People can sink in any environment. For example, I sank at Duke in a certain sense, but it's
Mhm.
not their fault.
Yeah. Yeah, yeah.
It's my own. But yeah, I think we have enough. Um, what happens is you get kind of a track record of doing things that are, um, people think are impossible or useless or both, and then pulling it off, and then the sort of people that would like to take that risk come,
Mhm.
and in practice, it's not as risky as it looks. I mean, that's the whole point, is we're not actually trying to pick things that where you're likely to sink. We're trying to pick things that where it looks impossible, but it's actually very easy. Um, and and just if we keep doing that, then people eventually get the message that somehow we figured out how to pick those projects that look hard and are actually easy. Um, so I think that's the main thing. We also have kind of a culture where failure is an option, not of the sink and swim type that you were talking about, but
Yeah,
the
yeah.
but the micro level failure. So in other words, instead of trying to spend a year designing the perfect experiment that works first time, I think some labs can do. Uh, we will do 1 million experiments in a day, and and we'll only get the top one or two that are the best ones. And you could say the rest of the 1 million are failures, but it's not like somebody's career is failing. It's that just 1 million experiments are failing in a day. And then the next day you do another 1 million, and each day you're taking the best of those 1 million. And so the best of 1 million, I think, is better to me than getting one perfect experiment to work first time.
No, for
There
sure.
you don't learn as much. The reason it worked is because you worked really hard on making sure that you weren't taking any risks. Well, if you do 1 million experiments, and this might be AI design a library, and then you screen the library. We made that possible in 2004 by being able to synthesize billions of sequences on a chip.
Yeah, yeah, yeah. It's just, yeah, it's just crazy to think, like, yeah, some labs, even I would say in the Netherlands, in Europe, it's kind of a common issue that, like you have to do a lot of preparations and stuff like that. Yeah, because probably, yeah, like there's a bit of a lack of funding, or yeah, not as, yeah, you don't have enough courage to do so. But yeah, it is indeed a common issue that, like, a lot of scientific ideas, they're just not being brought up to the next stage because of the, yeah, because of that. Yeah, so also Wyss Institute is very is a very nice place if you want to commercialize your research, and you have actually, alongside your professorship career, you co-founded around more than 50 biotech startups. So each time you come up with a new research idea, do you already have in mind the idea how to bring it to the market or commercialize it?
Well, I think we have the same level of attraction to kind of pure science topics as everybody else. Um, but we also are attracted to thinking about the societal benefits at the same time, and also safety issues and a variety of engineering issues related to that. And so in a certain sense, we have an integrated project from the very beginning where we're doing basic science and tech development and societal outreach and considerations and safety and ethics and all that wrapped into the beginning.
Mm.
And, it used to be you'd have to pick one. You couldn't do them all at once, but with these new technologies, you really can have it all. And so yes, to your question, it's relatively easy to make the transition into industry because we've picked the projects so that they are ready
Translatable.
for that transition. Yeah.
Hmm. Hmm. Yeah, yeah, that's yeah. And also, on this note, some scientists focus on, like, specific biological questions, while you yeah, like throughout your career, you focus more on creating tools that open up even more questions to ask. So, and yeah, as you said, as we already talked before, your lab is very famous for developing different technologies, like different sequencing technologies, multiplex genome editing, and yeah, and some other molecular methods. So, do you think that the technology development is underappreciated in academia, relative to just the single discoveries. Because we don't really think, right? Like we use all of these tools, but we in the back of the mind we don't really think that like how valuable they are.
Yeah, I think that's definitely what you described is definitely true. It's not necessarily a bug. I mean, it could be that you don't want everybody in society uh, working on radical technology development,
Yeah.
and it's definitely not doesn't fit a lot of people's personality because like with tech development, with clear hypothesis testing with very well tested methods, you can come in every day and expect the experiment to work. It may not give you the answer you wanted, or biology might surprise you, but that's usually a good thing if you have an open mind. But every day, it's going to work, right? Especially if you have all your machines backed up and lots of good technical help. But with technology development, you can count on almost every day it's not working because it didn't work yesterday and it's not going to work tomorrow. Eventually it will work. And so I think that's a different personality for one thing. But, in places, like my lab and the Wyss Institute, you get enough support and you kind of have the million failures a day attitude, and you've got, really cutting edge machines. It took the world, after our 2004 paper, it took them about a decade to start using DNA from chips. Wasn't even in review articles where they were talking about DNA synthesis. They were just discounted. It's as if it didn't happen. It was published in Nature. It's as if it had been published in the most obscure journal ever. The only people that used it were people that knew us, that were collaborators or stuff like that. So that was a decade that we had the advantage that even though it made it public, we were the only ones using it, and so we got used to doing libraries and using AI. It was a perfect match for AI. In fact, it's one of the few good matches for AI. I think ChatGPT and even folding of proteins isn't nearly as useful as being able to synthesize new proteins with new functions and go. I think that's the big payoff for AI so far, in my opinion, and our libraries were just ready for it.
Yeah, and also continuing a bit about the AI projects, because I know that you have co-founded some companies also that were focusing on AI drug development. Were these also like some kind of AI protein models that you explored, or yeah, what kind of AI tools do you usually use actually in your research?
Yeah, well, the most productive ones so far are large language models where we can design a key part of a protein machine, like a capsid of a viral delivery system, or ribosomes, ribosomal synthesis, very complicated systems. They're some of the companies we spun off are Dino and Manifold and Nabla, and these are making their way into clinical trials. You can do again multiplexing, but now not just at a molecular level, but at a whole organism. So we can design with AI a million different viruses, let's say, a million different capsids for gene delivery, and then inject that into a primate. And instead of doing a million primate experiments, which would be extraordinarily prohibitively expensive, you can now get a million at once in one primate, and then you can repeat it and you can change it and do cycles of iteration. So that's a whole nother thing that could greatly shorten our clinical trials and improve. We've improved the specificity of, say, our delivery by 100-fold, or say towards the brain, and moved it 10-fold away from the liver where it goes too much. And you just imagine all the other experiments you can do that by that combination of AI plus large libraries plus getting as close to human as possible in the first experiment. So rather than working your way up from in vitro to bacteria to yeast to mouse to and then finally to,
Mhm.
to primates, go straight to primates.
Great. Mhm. Yeah, 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 yeah. It's just insane how many things are getting solved now with the use of AI. And but also talking about your other companies, you're also a co-founder of Colossal Biosciences. Yeah, like as we previously mentioned, the company that aims to de-extinct lost species like the woolly mammoth, Tasmanian tiger, and the dodo, and some a lot of other projects. And yeah, this company has certainly captured the public attention, but also it was significantly criticized. Some people argued that the technical challenges are enormous and the de-extinction is not as important as actually can how do you say maintaining the ecosystem, the current ecosystem, and and the benefits of the de-extinctions are unclear. So what would you say is the ultimate goal of these kinds of de-extinction projects that Colossal does?
Yeah, I mean, first of all, I have to say that almost every one of my ideas have been criticized.
Yeah.
Next-gen sequencing and CRISPR and the pigs transplants and you name it. So this is not unusual. Uh, so basically, it's criticized until it works, and then oh yeah, that was obvious. Yeah, yeah. Let's move on. And so I think that. And it's also a misunderstanding uh, that we're just looking to de-extinct species. We're mainly looking to de-extinct alleles, meaning
Mhm.
individual gene variants that have some advantage for modern species. We're trying to, restore and rejuvenate specific environments, like the Arctic really should stay frozen. Okay,
Mhm.
we're in favor of keeping it frozen in the wintertime at least. And when I was just last in Siberia, it
Mhm.
was the first year on record that the summer thaw did not get refrozen in the winter thoroughly.
Wow.
So yes, we're definitely interested in current environments. That's our top priority. Current endangered species, various endangered wolf, elephant, and rhino species.
Mhm.
Uh, And whenever we can do two at once, so that's great too. So if we can save an endangered species and have some impact on climate change, as we're trying to do with cold-resistant elephants, but we don't need to restore every single gene.
Mhm.
In fact, in many cases, we want to put in new genes, like genes that
Mhm.
cause resistance to herpes virus, which is killing off the infant elephants. Um, so it's really a very pragmatic thing. If we will develop technology where we could replace the whole genome, but it's not a priority. And to criticize us for why are you dinking around with Earth orbit when you could be going to Mars or to Alpha
Mhm.
Centauri? Well, that's
Mhm.
not a fair criticism because in Earth
Mhm.
orbit you can do GPS satellites and telecommunications and weather, and those are perfectly valid intermediate things to be doing. Maybe
Mhm.
even better than the long range things. So anyway, that our focus is on endangered species and current um, restoration of the and maintenance of our best environments.
Yeah, that's what we yeah that's what I also got. I think that's the main mission. But also in Europe, yeah, like we have the ethical considerations have a lot of say in regulating like the new stuff that comes up in science, and not only in science but also in the industry. So from your perspective, what is the right way between balancing the necessary caution and between science moving forward? So How do you think? How
I,
can we
I'm
do?
I'm always in favor of safety first,
Yeah.
because in fact I think safety is a valid engineering principle. Almost every field of engineering has, like the plugs that you stick into the wall are, been designed to be as safe as possible, and bridges and buildings aren't supposed to fall down. It's things like this. There's a whole discipline of trying to improve safety, and nothing's perfect. A million people die in automobile accidents, so clearly that safety could be improved. But
Yeah.
but that I think that's one of our major engineering goals, is for safety, and certainly doing nothing is not a great option when the Arctic is melting and it's releasing carbon dioxide and methane. Uh, that's not a really great option. If
Mm.
somebody has a better idea, where they can do it entirely mechanically and that's
Mm.
actually cost effective, and then I'd be the first to be supportive of that. Um, uh, Also, I think there's general, you can rewild, as I say, you
Mm.
can bring animals back, um, that are engineered by breeding. I mean,
Mm.
that's something we've been, or you can do it by changing one base pair with, CRISPR or something like that. So, and you can't tell the difference. Something where you've done the cross by breeding and where you've introduced the small change. And in fact,
Yeah.
if anything, the CRISPR is more precise rather than getting, the reason that people were worried about the GMOs is we're bringing in a whole package of DNA from other organisms and you didn't really know what was going to happen. But now they're saying, well, let's just do random breeding where you don't really know what's going to happen as opposed to point mutation. So I think
Mm.
people are starting to warm up to this
Uh-huh.
idea, um, in cases where the benefits are clear, uh,
Mhm.
which I think there are a lot of cases where the benefits are clearer than they were for food. Like the salmon, for example, the
Mhm.
engineered salmon was not clear how society benefited from that. But
Mhm.
eliminating malaria, eliminating, reversing carbon are very clear societal benefits.
Yeah, definitely. And also for the so to say moonshot project of the Colossal, like the um so to say the mammoth de-extinction project. It's all as you said, right? It's wound up on the minimal genetic engineering. So like you tweak maybe 6, 7 genes, like in the elephant.
69 or
Oh, no,
609.
no, no. Yes, 69 or 79. More in the case. Yeah, in this case. In order to get something that is like as close can be as close like by its physical appearance and the traits to the mammoth as possible. But do you think actually um for the future, releasing this to the wild could benefit the ecological situations in any case, or it bring even more problems, so to say, right? Like the shift of the um current ecological like ecosystem or something like this, or do you think that it has more benefits?
Well, it's not for me to have an opinion on, it's something to do experiments on so we know the answer. So, for example, let's say you've got a desert that's spreading faster than we would like, or we would like to restore it to a previous state. You can do
Yeah.
experiments on the edge of the desert and see whether, you know, um, new plants. They could either be old plants that died off, or
Mm.
only present in seed vaults, or they could be new hybrids and so forth. You don't even have to use recombinant DNA. You can do experiments to see um, if it works or not. And I think there's going to be lots of that. I think there's going to be
Mm.
lots of cases where previous solutions and some new ones are very attractive. But it's not going to be based on first principles where you just say, oh, recombinant DNA won't work and breeding will, you'll try things and you'll be very cautious
Yeah.
about it. The way you are with drugs, is we
Yeah.
don't
Yeah.
we don't just think oh, we're gonna cure measles without vaccines. That's a popular thought. You don't just do it ideologically, you do it
Mhm.
experimentally.
Mhm.
And many of these things we have to learn the lesson over and over again.
Yeah. 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 That's true. That's true. Okay, and talking on the other note, in recent years, you've ventured into biotechnologies aimed at reversing aging or slowing down aging. And like in your lab, there has been multiple experiments on mice that basically you shoot one shot of the drug into the mice, and it could it could address multiple aging issues after only one shot. And you also co-founded Rejuvenate Bio to apply these approaches. So um, in some of the interviews, you mentioned that you think therapies that slow aging or even partially reverse it will become available in the near future. But also when I spoke to we even had a course, like a separate course, like on the ethics of science and ethics of technology, something like that. And like some people think that if these therapies succeed, we may accidentally create like a full new class of humans, like superhumans, like with longer lifespans, fewer diseases, maybe even enhanced capabilities. While other people that are not reachable to this technology, they stay normal, so to say. So, and how do you think if these therapies they become available, how do we make sure that this doesn't um, widen the inequality between people and just um, basically not separating the humans into
Yeah.
superhumans and the normal humans.
Yeah, I think that aging is a really good example. Part of the reason I'm attracted to it is the possibility we could get very low cost and very high quality.
Yeah.
Tends to be true for the drugs or the technologies that affect the largest number of people. So it's like you have a kind of a fixed cost of research and development and clinical trials, and then if you have a very small number of people in the denominators, it's maybe 100 people a year for rare
Yeah.
genetic diseases, um, then it's going to be very expensive, and it's going to be the rich benefit differentially. And I think this is true for, rare
Mhm.
diseases are better handled with genetic counseling, where you're talking about hundreds of dollars rather than millions of
Mhm.
dollars. But
Mhm.
for aging and pandemics, there you billions of people could use it, and so the cost for gene therapies, for example, or protein therapies, and
Mhm.
any of the orphan drug type things, when they now become a common drug, change from millions of dollars to $30, and
Mhm.
that's something that even if the individual can't afford $30, the government it's a no-brainer because you're going to extend their useful years, their healthy years, and you'll get that $30 back,
Mhm.
thousandfold over.
Mhm.
So it isn't not every technology makes the gap bigger. So for example, smallpox is something where literally all 8 billion people benefit. They don't have to get shots, they don't have to get drugs, they just benefit because we made it
Mhm.
extinct. And so there are lots of technologies like that. And I think aging reversal will be one of them where everybody should be getting the same benefits if they
Mhm.
want them. Now, there's still going to be people that don't want the technology, and that should be their right. But certainly education and discussion, you know,
Mhm.
dialogue should be part of to make sure that they know what they're not asking for.
And also, we talked about the germline editing. So, what do you think is the right way in which we can use the germline genome editing? Because it's also like a very, yeah, like it's a very big ethical thing. Yeah, like even in comparison with somatic genes editing, it just
Well,
stays with you.
I think the main problem with germline, so very often when it's framed ethically,
Yeah.
so I write a fair number of ethics and safety related articles, but most of the competing articles, they're not suggesting solutions and what's the barrier to those solutions, and they're often talking about it in very vague terms like this is icky
Mm.
or I don't see how we could do this. Um, I think the main problem with germline is that it takes a long time. So, um, especially if you're aiming for some late onset disease like Alzheimer's, that's a
Mm.
bad combination. Not just because it's unethical, but because the reason it's unethical is because it's hard to do the test where you show you treat somebody at age 0 and then you
Yeah.
wait 70 years to see if they actually don't get Alzheimer's. Now, very early onset things where you can't cure them with orphan drugs like microcephaly, for example, where the brain is incredibly tiny, that's something that would make more sense than Alzheimer's. But even that has alternatives like genetic counseling. Or
Mm.
if you did IVF, you can detect a lot of these things in an earlier stage.
Mm.
So I think the second thing that's wrong with gene therapy, somewhat related to that, is that it's uh, for germline gene therapy,
Mm.
is that 8 billion of us are excluded from that. 8 billion of us have already passed the point where, and in general, we don't want to have things where the drug is excluded from a major part of the population. In this case, it's the entire population. So I think
Mm.
that's a rephrasing instead of putting it in ethical terms, of putting it in medical, practical terms.
Yeah.
So the place that where might become valuable, an enhancement is, we are enhanced. I mean, we're
Mm.
relative to our ancestors, we're not in fear of most pathogens. Um, thanks to vaccines and antibiotics and antivirals.
Mm.
So I think there will be things like that
Yeah.
once we get used to the somatic gene therapies, you'll move them up earlier and earlier. Um, I think it's just not wise to say something is categorically not going to be tested. So, for example, some countries, for example, the United States, doesn't even allow us to go to the FDA with good findings. I think that will change as soon as we have good findings. And in the meantime, we can do germline via pigs. If you want to get the benefits of germline, do it in pigs and then transplant it in the humans, and that's all legal. Um,
Yeah.
so I think it's really impractical right now. Nobody has really laid out a specific disease they'd like to fix. It probably should be something that affects all of us to get the price down and to eliminate things that you could do with genetic counseling.
Yeah. Yeah.
Yeah,
And
I unfortunately have to go
yeah.
now. I'm a little late for my
Yeah.
11.
Yeah.
Yeah,
Oh, yeah. Sorry about that. Yeah, I just completely lost track of time because it was
I'm
very
fine.
interesting. The conversation, the
Yeah,
conversation was amazing, and yeah. Thank you very much again for sharing all of
yeah,
the insights with us, and yeah, we
okay.
we I hope we stay in touch.
Yeah, okay.
Sure.
Take care.
Thanks for listening to the new episode of our interview series. If you enjoyed it, don't forget to like, subscribe, and share it with your friends. See you in the next episode.